Layered double hydroxides, LDHs, are a class of layered materials with a general formula of Me 1 minus X, Me X O 2 X plus and minus bi per meter NH2O, and a structure. The layers contain divalent Me and trivalent Me metal ions, and the interlayer region is occupied with charge balancing anions. Divalent metal ions are often Ca2+, Mg2+, Fe2+, Co2+, Mn2+, Ni2+, Cu2+, or Zinc2+. Trivalent metal ions are commonly Al3+, Fe3+, Co3+, and Ni3+. The anions, such as CO32-, NO3-, and Cl-, in the interlayer galleries can be readily replaced. Therefore, drugs may be conveniently loaded to LDHs simply by ion exchange. By loading drugs in the interlayer regions, LDH is capable of protecting the drug molecules. LDHs have very large specific surface area, and therefore drug loading quantity is high. LDHs at nanoscale are readily dispersible in aqueous media. Furthermore, LDHs are positively charged while cell membrane is negatively charged, which is beneficial for enhanced cellular uptake. These advantages have been demonstrated in extensive studies of using LDH nanoparticles for delivery of a range of drug molecules. In one study, Mg 0.68 Al 0.32, O2, NO3 0.321.2 H2O, Mg2 Al NO3 LDH, was firstly prepared by a coprecipitation of mixed metal ions, Mg2+, Al3+ equals 2:1, in a base solution, 0.1 NNaOH, pH approximately equals 10, under nitrogen, 51. Different materials were then loaded to the interlayer regions of the LDH nanoparticles. Before loading of these materials, the basal spacing of the LDH nanoparticles was 8.7 A. After being loaded with herring testis DNA, adenosine triphosphate, ATP, fluorescein 5-isothiocyanate, FITC, and CMYC antisense oligonucleotide, as MYC, through simple ion exchange reaction, the spacing was enlarged to 23.9, 19.4, 18.8, and 17.1 A, respectively, calculated from X-ray diffraction results. FITC-loaded LDH nanoparticles were used to perform cellular uptake study, and the results revealed that the uptake of the LDH Fitch hybrids was well enhanced with increased concentration and incubation time. Confocal microscopy observation showed that the fluorophore was detected in cells within 1H of incubation, and the signal continuously increased for over 8H. The fluorophores in the cells were primarily distributed in the peripheral and cytosol regions with some in nucleus. This was in line with the TEM observation in a separate report. For the cells treated with LDH Fitch hybrids at 1 μm concentration, fluorescence could be observed in cells. In comparison, for the cells treated with 5 μm FITC alone, no fluorescence signal was observed inside cells. It is evident that LDH helped the intracellular uptake of FITC molecules. In another study, gamma-32P, labeled ATP, was employed to conveniently compare the cellular uptake efficiency of ATP molecules alone and LDH-ATP hybrids. The experimental data clearly verified that greatly enhanced transfer efficiency could be obtained for LDH-ATP in comparison with ATP alone. Following the confirmation that LDHs function in aiding intracellular delivery, LDH nanoparticles and LDH as MYC hybrids were incubated with human promyelocytic leukemia, HL60, cells. At a concentration below 1 mg per milliliter and incubation time for up to 4 days, LDH nanoparticles did not show noticeable cytotoxicity. In contrast, at a concentration of the hybrids of 20 μm and incubation for 4 days, 65% growth inhibition of the HL60 cells was observed when compared to untreated cells. The use of LDH nanoparticles for improving DNA vaccine delivery in the treatment of melanoma in mice was also recently studied. The results demonstrated that with LDH nanoparticles, the DNA vaccine could induce well-enhanced serum antibody response compared with naked DNA. With the success of using LDH nanoparticles for DNA delivery, the application was extended to anti-cancer drug delivery. MTX is widely used for human cancer treatment. However, MTX has a very short plasma half-life and therefore it is often used at a high dose, which may lead to drug resistance and non-specific toxicities in normal cells. Researchers have carried out studies attempting to use LDH nanoparticles to solve this problem. MTX was hybridized with LDH nanoparticles by ion exchange reaction. Before MTX loading, the basal spacing was 8.4 A. After loading, the spacing dramatically expanded to 19.9 A. This indicates that MTX entered the interlayers of the LDH nanoparticles, which was further confirmed by comparing the FTIR spectra of the LDH-MTX hybrids, LDH nanoparticles, and MTX. The samples were then tested in one normal cell line, human tendon fibroblast, and another cancer cell line, human osteosarcoma SAOS2. The results showed that LDH-MTX hybrids, MTX, and LDH nanoparticles did not have significant effect on the proliferation of normal cells. However, LDH-MTX hybrids and MTX suppressed the proliferation of tumor cells while LDH nanoparticles still did not have effect. When comparing the drug efficiency of different samples at various periods of times, it can be concluded that, during the first 3 days, LDH-MTX worked much more efficiently than MTX. The mechanism was investigated by extracting genomic DNA from the cancer cells after being treated by MTX with a concentration of 500 micrograms per milliliter and carrying out electrophoresis analysis of the DNA. In the cancer cell group treated by LDH-MTX hybrids, internucleosomal cleavage by DNA apoptosis was observed while no noticeable DNA apoptosis was detected in the groups treated by individual LDH and MTX. Overall, the study suggested that LDH-MTX hybrids damaged the DNA of cancer cells more efficiently than MTX and correspondingly had well-enhanced drug efficacy. The reason may be as follows. First, LDH protected MTX from rapid decomposition in blood flow. Second, LDH increased the cellular uptake of MTX by cancer cells. LDH nanoparticles were also used for siRNA delivery. It was demonstrated that LDH-mediated siRNA delivery effectively silenced neuronal gene expression. Except aiding in intracellular delivery of a range of molecules, LDHs were also used to reduce harm of drugs to patients. Indomethacin is an anti-inflammatory drug with very bad gastrointestinal side effects. It can cause peptic ulcers, which may result in serious bleeding and perforation. To eliminate or at least reduce the side effects, the drug was incorporated with LDHs followed by in vivo study in mouse model. The results showed that the LDH indomethacin hybrids caused much less gastric damage compared to oral intake of the drug alone. Furthermore, controlled and sustained release could be realized by using LDHs. In one study, various drugs including antibiotics tetracycline, DOX, 5-fluorouracil, vancomycin, VAN, sodium fusidate, SF, and antisense oligonucleotides were bound to LDH clay particles and then dispersed in lactic co-glycolic acid films. The control groups were drugs bound to only LDH clay particles. Without lactic co-glycolic acid, the drugs showed a large burst phase of release, except DOX, with little extended drug release for 4 days. With the polymer films, the drug systems had a reduced burst phase of release followed by a slow continuous release for over 12 days. Very recently, another study in rabbit model was reported in which LDH nanoparticles were impregnated with antibiotic ciprofloxacin and coated on BioVerit-2 implants to act as a sustained drug delivery release system against an infection with Pseudomonas aeruginosa in the middle ear. The results indicated that LDH nanoparticles may be very useful for controlled release applications because they are capable of protecting drugs from being degraded and releasing effective drug doses over a long time periods.